Hi everyone and welcome to BSC 1005, which is Biology for Non-Majors. My name is Dr. Horman and I will be your instructor during the duration of this course. As we peruse through our different chapters, if at any time you have a question, a comment, or a concern, please do not hesitate to reach out and I will get back to you as soon as possible. Today's lecture is going to focus on Chapter 1, which is entitled The Scientific Study of Life. As we peruse through our chapter, we're going to explore a few different topics. We're going to start off by taking a look at some of the characteristics that scientists consider to be important for an entity to be considered alive. We're also going to take a look at the scientific method, which is a series of steps that scientists follow whenever they're conducting research. And we're also going to start exploring the difference between a prokaryotic and a eukaryotic cell. Now, speaking of cells, the cell is considered the basic unit of life. And the reason that is so important in biology is because, by definition, biology is the scientific study of life. And the cell can be found in every single organism, whether we're looking at a unicellular organism such as a bacteria, or a multicellular organism such as a plant, a fungus, or an animal. One of the reasons why the cell is also so important is because that is where we're going to be housing our DNA, our deoxyribonucleic acid. Here's a nice little image of it. Now, this little double helix contains our genetic code. It contains all the information we need to produce various proteins, as well as some of our behavioral and physical attributes. As we explore our different chapters, we will talk more about the overall structure and function of DNA, and we'll get to learn exactly how it does produce those particular proteins. But for now, let's go ahead and take a look at these characteristics that I've mentioned to you before. Now over here, this is table 1.1, and as you can see, there are 5 characteristics listed that we should see at the very least a combination of whenever we're pursuing or trying to establish if an organism is alive or not. So over our next few slides, we're going to take a look at these characteristics and find out exactly what they mean when they say things such as organization, energy use, internal consistency, reproduction, and of course, evolution. Let's go ahead and start from the top and work our way down. So our first characteristic is that if something is alive, there should be some type of organization within it. An organization often pertains to the fact that there is a hierarchical pattern, a way to break the organism down into smaller pieces, or you can think about it as starting off very small and building upon it. And oftentimes when you do that and you start with a building block and you continue to build and grow upon that, we start to see that not only do you have that hierarchical pattern, but you'll also have emergent properties. And emergent properties basically means that the sum is greater than the individual parts. So by being organized, the entity will get a greater benefit out of the process versus just staying at a simplistic level. Let me give you an example. This is image 1.2 in your book, and it basically shows you how we can see our different levels of organization in a biological sense. Now, in the center of the image, you can see there's this beautiful tree, and this is going to be our organism. Now, you can also imagine a beautiful image of yourself right over here, because you would also be a perfect example of an organism. Now, how can we demonstrate our hierarchical pattern, our level of organization? Well, we can go 2 ways. The first way we can go is that we can take a look at ourselves and realize that we are a large entity that is the product of several smaller parts. So we're going to follow this trail right here. And as you can see, we're going to start off real simple by taking a look at things like carbon, hydrogen, oxygen, and nitrogen, which are all different types of elements that compose our body. And these atoms are able to follow a hierarchical pattern and start increasing their size and complexity by pulling together and forming a molecule, like our DNA. And that DNA can then be housed in different organelles. Now, for us, we take a look at our nucleus, but for plants, we also find some DNA in, for instance, their chloroplast, which they then use as part of their cell. Remember, that's the basic unit of life. And if we have cells, we can make tissues And if we have tissues, we can make organs. And from there, we just keep building until we have an organ system, of course, which of course brings us to the organism. So as you can see, we started off small and we ended up larger, more complex. But we can keep going because remember, we don't live on this earth by ourselves. So we are part of a population, a group of similar species and organisms that live together. So that becomes more complex, which then allows us to be part of a community, an ecosystem, and eventually a biosphere. So we can go as extreme as we'd like when we take a look at our hierarchical pattern. Now, another thing that I mentioned to you before is that as you start becoming more organized, you will have emergent properties. And emergent properties is basically the fact that these individual pieces, when they start interacting, not only do you create something that's larger and more complex, you'll also get a greater benefit out of it. So the perfect definition of emergent properties is the sum is greater than the individual parts. And this example right here is a nice one. So here we start off with some endothelium cells, which are just some abstract shape cells. I think many of us will think endothelium cells. An example of that would be our skin cells. So they're very thin, they're very flat, they almost have like a scale-like appearance, and they're very simplistic, right? So what we can do is we can take these cells and we can start merging them together. And as you start growing and adding more of them together, eventually you get a sheet, so you become more complex. But we're not going to stop there. We're going to keep growing, and eventually the sheet of cells will get so large and complex that it will actually fold over and create the appearance of what looks like a little cylinder. And your body can now use this more complex structure for a very great function. And in this instance, it's going to use it to transport our blood vessels. I'm so sorry, our blood cells. So here you can see your capillary and you can see our little red blood cells because our endothelium cells have laid the basis for transport of our red blood cells as well as our white blood cells and our platelets. So as you can see, this is an example where you started out very simplistic and now you are larger and more complex and you get a greater benefit out of it, aka emergent properties. The second characteristic to determine if something is alive is to see if it requires energy. Now, energy is the ability to do work in the scientific sense, and for that we often reference our metabolism, which takes a look at how our body is able not only to break down energy, but also how can it acquire energy. And it turns out that there are two options. You can have organisms that are labeled as autotrophs, meaning that they're able to produce their own food source, their own energy. So they're called producers. Or you can have a consumer or a heterotroph, which relies on other organisms, other autotrophs or other heterotrophs to acquire that energy. Let me give you an example by our little food web that's illustrated for us over here. So let's start our story with the sun. The sun is nothing but just a ball of energy that's radiating on our earth. And it turns out that some organisms can utilize that energy and extract everything they need out of it to produce all their nutrients. Those organisms would be called autotrophs, and an example of that would be the plants right over here. Another example of an autotroph would be a cyanobacteria, a bacteria that can photosynthesize, very similar to the plants, and able to produce all their own foods. Now, as you can see, the plant is minding its own business, producing all its own food, when this little beetle comes by and starts chewing on it. And what the beetle represents is a consumer or a heterotroph. The beetle cannot extract its own energy and make its own nutrients from non-living environment. Instead, it's going to have to consume it from a living source, such as the plant, and thereby allow that energy to transfer from the plant into its own body. This is the same thing we do whenever we sit down and we have meat products or plant products. Part of why we're consuming that is so that we can extract the energy from their cells and allow it to enter into our bodies. So we are a perfect example of a heterotroph. Now, we're not done yet because eventually we'll see that the organisms will become deceased. And we still have energy harvested in those carcasses or dead organisms and in their waste. So that will then be the job for what we like to call decomposers. Decomposers are a specialized, or I should say a subgroup of heterotrophs, which exclusively feed on dead organisms and organic waste. And they are essential to our food web because they allow the carcasses to be recycled and the nutrients to be returned into the soil so that once again they can enter the food web and be used by producers to extract additional food from it. And it's this continuous cycle that just keeps on going. So remember, if you're looking at an organism that's able to extract their energy and make their own food, then we're looking at an autotroph. If we're looking at an organism that has to consume other organisms to get their energy, then we're looking at a heterotroph. Our third characteristics for if something is alive is that there needs to be a level of internal consistency, or better yet, there needs to be what they call homeostasis. Homeostasis is centered around the fact that certain parts of our body are limited to a set point. So for instance, our internal temperature, our internal pH, it has a particular set point where the cells are best adapted to it. And part of what homeostasis does is that regardless of what happens to our internal or our external environment, we must do everything we can to remain within that range of the set point. So let me give you an example. So I like to talk about body temperature a lot when we do homeostasis because I think many of us can relate to examples of things like sweating or shivering. So we know what happens when we get hot or when we get cold. Now, our internal body temperature works best at 37 degrees Celsius, which is around 98.6 degrees Fahrenheit. And if you look at your chart right over here, what it's trying to tell you is that for us, homeostasis sets our internal temperature right over here. So this is our set point. Now, does that mean that throughout the entire day, that if we were to take your temperature, that you would always be at that perfect 37 degrees Celsius? Nope. Because homeostasis says, yes, you have a set point, but you are able to fluctuate a little bit up and down as long as you come back to the set point on the average. So what we see happening is throughout our day, as we expose ourselves to different environments, we can increase or decrease our temperature. So for instance, let's say it's really hot outside and you go for a walk. That's one of the reasons why you can start increasing your body temperature. Thank you to the sun. And as we go inside, for instance, maybe it's really cold, the AC is set at a low temperature. We start to see that we lower our body temperature because we're cold. Now, how does our body react to that? Well, many of us know that if we get too hot, we start sweating because that is the body's mechanisms to start cooling you down so that you don't overheat and return back to your set point. On the other hand, if you're cold, you might find yourself quickly shivering. And part of that is because your muscles are contracting and relaxing so that they can generate heat, almost like a warm little blanket that will surround you and warm you up and return you back to your set point. So internal consistency is homeostasis. And once again, keep in mind that homeostasis simply says that there is a set point that we function best at. We are able to fluctuate a little bit up or down, but we should always average out right around our set point. The fourth characteristic is that if something is alive, there should be some evidence of growth, development, and reproduction. And reproduction is essential because remember, biology is the scientific study of life. So they need to have a way to, in essence, allow for a next generation to come to flourish and see how the DNA and the cells change from one generation to the next. Now, reproduction can be done either through asexual reproduction. A means without, so there is no sex involved. Instead, you have one parent that can go ahead and make an identical copy of itself and pass that off for the second generation. So here, for instance, we have some bacteria and we have some yeast cells, And what we see happening here is that this one bacteria cell is splitting itself in half. So it can do this thing called binary fission, allowing for the daughter cells to form. And here in the yeast cells, we see these little buds. These are little copies of its DNA that it's basically releasing from the parental cell to create new cells. Now, whenever you only have one parent involved and you do asexual reproduction, that will mean that all the offsprings will be identical because there is only one source of DNA. On the other hand, when you do sexual reproduction, this involves two parents, which means that the offspring will include a combination of both of their DNAs and both of their traits. This is fantastic because it allows us to create lots and lots of genetic variety. And the more variety you have, the larger the chances are of survival. And more importantly, the larger the chances are that your offspring will be able to have offspring of their own. Here are a few examples of reproduction at work. So in our first image, we see some fungus that is reproducing through their spores. This would be an example of asexual reproduction that they tend to utilize. The spores are released in the atmospheres, and whenever they find a location they can grow on, let's say for instance, maybe you had some fruit or maybe some bread on display outside and you didn't put it away fast enough, you might notice that there is like a fuzzy little growth that occurs on it. That is because the spore landed on it and started reproducing asexually. Plants can produce both sexually and asexually. It will depend on the plant that we're looking at. And for many of the animal species, they rely on sexual reproduction. However, there are a few that can do asexual reproduction. A lot of times we'll have to look at the size and the complexity of the organism to see if asexual reproduction is an option for them. Our last characteristic is that if something is alive, there should be some forms of evolution. Now, go ahead and take a look at the definition of evolution. It says it's a genetic change in the makeup of a population. What this means is that as the DNA is passed on from one generation to the next, over time there are minute changes in the DNA, and as the DNA continues to change, there will come a point where you give rise to a new species. Evolution does not indicate that you were one organism on, let's say, Monday, and come Friday you were a different organism. It takes a look at how the organism evolves or changes over time. And part of that comes from the fact that genetically we have certain adaptations. And an adaptation is an inheritable characteristic, something that is embedded in your DNA that you receive from your parents. So some things, for instance, like your hair color, your eye color, your skin color. And what we see happening is that certain adaptations make it favorable for us to survive in the environment that we're in. And the more favorable adaptations you have, the more likely it is that you will find a mate. Because this thing called natural selection tells us that innately we aim to find a mate that will make it more likely for our offspring to survive and to thrive. Natural selection is often also referred to as survival of the fittest, and it basically says that if you live in an environment where there are limited resources, you will tend to seek out a mate that is best suited for that environment so that the chances of those favorable adaptations being passed on to the next generation is high, allowing for their survival rate also to be at a higher point. Let me give you a little example. Here is a picture of an adder snake, and as you can see, he is burrowed in the ground, and he is a perfect camouflage for the environment he's in. Now, his skin tone, or I should say the color of his scales, is determined by an adaptation. It's determined by its DNA. And as you can see, it allows him to have a favorable adaptation, which allows him to burrow in. And as he's perfectly hidden, it makes it easier for him to look for his next meal. Look at that delicious lizard he was able to catch. Now, the more successful the snake is at catching food, the more energy it has, which means the more likely it is to survive, and the easier it will be to attract a mate that will want those characteristics to be passed on to their offspring. So natural selection basically states that this would go ahead and warrant survival of the fittest. Now, is there such a thing as a perfect organism? And the answer is no. The reason for that is because the environment that we are in are consistently changing. And as the environment changes, the adaptations that are favorable one time can become unfavorable as the environment alters as well. So there's this constant struggle of which adaptation is favorable and what natural selection will lean towards. We also see a lot of evolution occurring, for instance, in our bacteria samples that are around. For instance, by us, we have an issue with MRSA, which stands for methicillin-resistant Staph aureus. Staph aureus is a bacterial infection that you would usually get on your skin, and you would kind of get little scabs or blisters, and you would go over to the doctor, and they would give you some antibiotics to kind of rub on it, and it would go away. Well, what we see happening is that Staph aureus went ahead and during one of its replication time had a mutation. Now, a mutation is a random change in your DNA. It cannot be predicted. And a mutation can be good, it can be bad, or it can be neutral. In the case for the Staph aureus, it was very beneficial because that mutation allowed its DNA to change enough that it was able to become resistant to a lot of the antibiotics that we have available. So that would mean that the antibiotic is able to kill off the old version here in green, but the red version is alive and continues to thrive. So over time, what you would see is that you would have an evolution of an antibiotic resistant bacteria. Once again, showing you how you have changes in the DNA that allowed for the organism to have different characteristics, aka a change in their genetic makeup, also known as evolution. Now, as we move through our chapters, we're going to be exposed to a lot of different organisms. And I think it's important for us to point out that a lot of times organisms will have a scientific name and an everyday name. So for instance, we are humans, but our scientific name is Homo sapiens. And when you look at the scientific name, it is provided by a branch of science called taxonomy, which is not only interested in naming organisms, but also classifying them. So trying to group them together in similar batches. When you look at a scientific name, what you're looking at is going to be the genus and the species name. And the genus name can always be considered almost like a last name. You're going to have a group of closely related species, whereas the species name you can consider your first name. It is you. There's just one of you. There's only one type of organism. So for instance, when we look at our scientific name, we see that we are Homo sapiens. And that is because the Homo for the genus hints at the fact that there are other organisms that came before us to which we were closely related. So for instance, the Homo neanderthalis or the Homo erectus. Now, the genus and the species names are a group of very closely related organisms. But as you can imagine, when you look at taxonomy and you look at classifying organisms, we can also start in very broad categories. And the broadest of them all will be what we call our domains. All living entities can be divided into 3 domains, and from there, they will continue to be subdivided into things like kingdoms, and from there, they will just keep going until they get to the more specific genus and species name. Here is a look of a phylogenetic tree, almost like a little family tree that shows us all the different organisms that we are aware of. And what this tree is trying to show you is that everything started with just one cell. That's that one cell. And as that cell replicated and reproduced, we started noticing some genetic changes in the makeup. And if the genetic change was drastic enough, it would start branching off from its original origin point. And as these organisms continued to change and evolve, it gave way to three domains. So these are the three large categories that we can put all living entities in. And those domains are going to be the domain bacteria, the domain archaea, and then the domain eukarya. The domain bacteria and the domain archaea are also classified based on their cell types, and they are what we consider prokaryotic cells. The domain eukarya gets its name because it is a eukaryotic cell. And if you take a look at the map, you're going to see some of the organisms that we're probably familiar with. That's because the domain eukarya will house our animals, our fungi, our plants, as well as what I like to call a little miscellaneous category called the protista. Here is just another look at the domain bacteria and archaea. Now, this picture, I would like to highlight once again the fact that they are classified as prokaryotic, and prokaryotic means that their cells do not have a nuclei. That just simply means that they don't have a central control center within the cell. The DNA instead will be free floating in the cell. And both of these domains are composed of unicellular organisms, meaning that they are just composed of one-celled organism. So a prokaryotic cell is a cell that does not have a nucleus, and it can be found in the unicellular organisms of the domain bacteria as well as the domain archaea. And don't worry, as we do more chapters, we're going to explore these domains with greater detail. Now, that's a prokaryotic cell. A eukaryotic cell is named for the fact that its DNA is housed within a nucleus. So now we have a control center. And the nucleus will often allow for a better level of organization. So we do see that the organism has the ability to become quite a bit more complex than a prokaryotic cell. And all the organisms that are eukaryotic can be found in the domain eukarya. And as you can see, there is a wide variety of organisms in here. You see your animal kingdom, you see your plant kingdom, you see your fungus kingdom, and then you have your protista kingdom. And the protista kingdom has a variety of both unicellular and multicellular organisms. So here we're looking at a little amoeba, which is unicellular. And then you will also find things like green algae in here, which is multicellular. The other kingdoms, the animal, the fungi, and the plant, those are mostly made out of multicellular organisms. But once again, all of them will have eukaryotic cells. As we reach towards the end of the chapter, I also want to talk to you about the scientific method. Now, the scientific method is a series of steps that scientists have to follow whenever they're conducting their experiment. And there are many different ways to discuss these steps, but I always like to say that it starts off with an observation. Something that kind of piques your interest that you want to learn more about. Now, for many of us, if we come across something that we haven't heard before or something we want to find out, maybe we want to know where the nearest restaurant is or what time the gym closes, we're kind of spoiled. We can take out our phones and we can do a quick Google and we'll come up with our answers. But the scientific method says, what if you can't find your answer out there or you find a partial answer? What do you do then? Well, it says you take all the knowledge that's out there and you come up with a hypothesis, an educated guess based on what you know, what you studied, what you found, what would be the possible explanation for the question that you're trying to answer. And the hypothesis doesn't have to be right or wrong, but it has to be testable. So oftentimes people will put it in an if and then statement. So if I take this medication, then the disease will behave this way. Or we can say it even more simplistic. If I go to the gym twice a week, then I can lose a little bit of weight. If I take the highway, I will arrive to my spot faster than if I take the inner roads. So it's all about giving an educated guess, trying to predict what the outcome will be, But more importantly, it will give you a base as to which you can build your experiment around so that you can collect your data and obviously publish your results and share it with the world. Here is a look at the scientific method in illustration. And what I love about this image is that it showcases it as a continuous cycle. And part of that is as you make your observation and you form your hypothesis and you start to set up your experiment, the goal isn't to create a end or a closed pattern. The goal is to publish your work and share what you've come up with, but more importantly, make it so that others can read about it and build upon that data that you presented. So if they knew the series of steps that you followed, aka if they know the scientific method, they can then go ahead and replicate your research or tweak it in different ways so that we can see any additional pros or cons that can be drawn from the knowledge that you are able to share. Now, when you set up your experiment, there are a few rules that we have to kind of follow to make sure that we're tailoring it to the scientific method. And one of them is that whenever you do an experiment, you want to make sure that you try to get a nice large sample size. And as you're conducting your experiment, you will often have a group that will be labeled as your control because you need to have a basis of comparison. Now, in your control, you can have a placebo, and a placebo means that you have an inert object or something that doesn't have any effect. So maybe like a sugar pill, or a lot of times you can have a placebo that is a treatment that's already out there on the market. Let me give you an example. Let's say we want to go ahead and we want to develop a new blood pressure medication. And in our blood pressure medication, we're going to add some new ingredients that make it different than the ones that are already on the market. When we go out and we test it on our patients, some of our patients will be the controls, and they will be able to get the medication that's already familiar with, that's already on the market. So that medication will then be our control or our placebo. You can also establish that your placebo will just be a sugar pill, meaning that the patients are taking the pill, but it absolutely has no effect on their symptoms. So then you have a true basis of comparison to the treatment group or the group of individuals that are taking the new medication. Oftentimes, your experiment will also be double blind. Double blind is a fantastic way to make sure that the data that's presented has no bias. And that basically means that the researcher, as well as the participants, do not know if they are part of the control or the treatment group. So that's why it's called double blind, because there are two individuals that are blind, both the patient and the researcher. Usually there is a third independent party that is hired that will assign numbers or identifying markers and will keep track of which group is which. The other thing I want to mention on this slide is when you set up your experiment, there are variables to consider as well. Some of the variables are going to be independent, and an independent variable is anything that you as a researcher get to decide. So you get to decide what disease you're going to study. You get to decide what type of medication you're going to develop. You get to decide the dosage, the quantity, the length of the medication. And then the dependent variable, that is the response of your treatment. How is the patient reacting? What is the outcome going to be? The dependent variable is not to be manipulated. It's simply meant to be recorded. Along the way, as you have your control and your treatment group, you will also have to establish some standardizing variables. And this is basically to make sure that any external factors, such as perhaps diet, water intake, exercise, are kept at equal levels across all groups so that you have your quote unquote standards. And that will then mean that if there is a clear difference between the control and the treatment group, it is due to your treatment option, not due to any type of external feature. Let's go ahead and kind of, here's table 1.2, my apologies. Gives you a nice little definition of the different variables. And what I meant to say is I want to take these variables and I want to apply it to the example that's in your book so we can see how these variables will work together. So I wanted to go ahead and show you this illustration right here. And in your book, it talks about the fact that there's going to be an experiment set up that wants to go ahead and it wants to test a newly developed vaccine against the rotavirus. And the rotavirus is usually caused due to contaminants in water. So you'll drink the water, it'll usually cause an upset stomach, diarrhea. For adults, it's just an inconvenience most of the time. But for younger individuals, such as babies, it could be detrimental due to extreme fluid loss. So coming up with a vaccine that will protect our immune system from this virus sounds like an excellent idea. Now, in the book, it talks about the fact that if you're setting up your experiment, you want to keep an eye on what you can control. So, for instance, as a researcher, you can control the fact that you want to study the rotavirus. You can control the fact that you want to create a vaccine. You can also control the fact that you only want to test it on babies. These options are all examples of an independent variable. So you got to pick it. You also got to pick the different groups, right? So as you can see, there was a placebo group, there was a low dose, a medium dose, and a high dose of the vaccine. Now, once the treatment is given, the response of the patients, how many were still exposed to the virus, excuse me, how many had a moderate or a severe reaction. Their reaction, that would be an example of a dependent variable because you can't control who's going to get sick and if they're going to get severely sick. So on the chart, the results that are showing in green, we're looking at the percentage of each group that showed any symptoms towards the virus, because remember, the vaccine will boost your immunity. It doesn't mean that you won't get sick. And then in blue, we're seeing the cases that have severe symptoms for the illness. Now, why are we looking at both of them? Well, we want to look at the ones that are severe because that will show us that our treatment option was ineffective because we weren't able to boost the immune system enough to provide us protection. So as you can see in the placebo, there's quite a bit of babies that were severely ill, and this can partly be due because the placebo in this case was nothing but just salt water, absolutely no effect at all. More importantly, if we take a look at our dosages, we see that even though you had incidence of illness across all three groups, so you can see the green chart in all three, the only group that had a severe case is the low group. Now, this means that our medium and our high vaccine dose were effective in protecting the kids because none of them were severely ill, but our low dose did not provide enough protection because we still had severe symptoms that made an appearance. Now, all of these reactions are bundled under the dependent variable. And in case you were wondering, well, what about the standardizing variables? Well, remember, these are your standards. So for instance, if you have your four groups, some of the standards would pertain to the fact that all the children would have a similar diet. They would have a similar amount of exercise. They would also be on a similar health plan and keeping it nice and consistent. So if there's any abnormalities, you know that it isn't because of malnutrition or dehydration, but instead due to the fact is whether or not they received the vaccine. The beautiful thing about science, and especially biology, is that we learn more each day. And part of that is because the scientific method tells us to just keep accumulating more and more and more data. And as this data becomes published, we're able to read it and share it. And of course, go back in and redo the experiments because the goal is to eventually go from a hypothesis, an educated guess, to a theory. And a theory in science allows us to have an explanation, allows us to understand items on a broader scope. We are still testing it, we're still learning more, but it allows us to have a better predictive power. So things such as the cell theory, things such as the germ theory, they are all out there because of little pieces of information that researchers have collected throughout the years, and we're able to combine it all together. So a theory is a wonderful thing, and it allows us to get a better scoop and a better predictive power. And one of the examples that I like to give of showing how useful a theory can be is towards the end of the chapter. And at the end of the chapter, when you get a second, make sure you read through the investigating life aspect. And it talks about Darwin. Now, Darwin is often known as the father of natural selection because he was one of the first scientists to say, you know what, organisms will benefit at certain parts of the environment, and organisms will go ahead and utilize their different adaptations, and they'll see traces of natural selection. Now, he didn't say it with all those words. Instead, he said organisms will descend with modifications. But along the way, Darwin started to learn about the wonderful beneficial relationships that plants have with different animals. And during his lifetime, he was roaming around Madagascar, the island, and he came across these beautiful orchids that had an extremely long nectar tube. And he thought that would be so odd that the plant would grow such a long nectar tube. And he went ahead and used all his knowledge to come up with a theory that stated that there had to be some type of organism that had a unique characteristic that would allow it to use that long nectar tube, draw out the nectar from the orchids, and also carry its seeds so that it can distribute it all across the island. And he looked high and low for this particular living organism, but he couldn't find it. But he did predict it and he wrote about it. And it wasn't until a few decades after his death that they discovered this beautiful moth right here, exclusive to the Madagascar island, which has a extremely long siphon that it's able to roll out and that fits perfectly inside the nectar tube. So it's almost as if the animal and the plant are symbiotically living together and benefiting from each other. Isn't that fantastic? And keep in mind, Darwin never saw this moth, but he had the predictive power of the theory because of all the research that he had conducted. Well, that is our last slide for chapter one. Please, if there's any questions, comments, or concern, please do not hesitate to reach out. And we shall talk soon, and I will see, or I guess I will speak to all of you again in our next chapter. Enjoy.